Stress behaviors and the gut microbiome of rhesus monkeys in a primate sanctuary. In rodents and humans, dysregulation of the gut-brain axis has been implicated in depression and stress, and probiotic administration can alleviate these effects. Primates in sanctuaries are commonly prescribed probiotics to reduce stress, but their effects on stress behavior has not yet been studied in this context. Primates Incorporated, a primate sanctuary in Wisconsin, aims to maximize resident well-being, and they had 5 adult male rhesus macaques that were prescribed a probiotic multivitamin that you can see here on the right by a veterinarian to promote well-being. We use non-invasive methods of behavioral observation and fecal collection at Primates Incorporated to explore relationships between the gut microbiome and behavior. Primates Inc. regularly collects 10-minute behavioral scans of each of their residents. We focused on stress behaviors, which include aggression, self-injurious behaviors, self-grooming, and stereotypical behaviors, and positive behaviors, which include foraging, drinking, moving, engaging with enrichment, and affiliative behaviors. We also collected feces and sent them to UW-Madison for DNA extraction and amplification. We then analyzed for diversity and abundances of 21 different bacterial classes and included the behavior from the 7 days preceding each fecal collection. We found that stress behaviors were negatively related to microbial diversity in the gut microbiome. One bacterial class, Firmicutes bacilli, correlated positively with both stress behaviors and with positive behaviors. Several other bacterial classes either negatively correlated with stress behaviors or negatively correlated with positive behaviors. After probiotic administration, the gut microbiome had an increased diversity, as you can see in the top left graph. The next graph to the right of that shows that the abundance of Firmicutes bacilli was also elevated after probiotic. This is to be expected because the probiotic included bacteria that were from this particular class. However, the 2 graphs on the top right also show that there were alterations to other bacterial classes that were not part of the probiotic. So the probiotic altered the ecology of the gut microbiome. Despite that, the 2 graphs on the bottom show that no behaviors were altered in response to the probiotic administration. In conclusion, diversity and abundances of bacterial classes correlated with behavior. Although probiotic administration altered the gut microbiome, it did not impact behavior. Non-invasive behavior and fecal collection in sanctuary settings are challenging to obtain, but they can provide valuable information to sanctuaries and the scientific community.